Hey guys, welcome to another episode of You've Got Potential, the educational segment. Today we're going to talk about spectroscopic methods. It's more like the physical sciences, but we're going to show an application in the biomedical sciences to sort of merge the two fields together. And there's going to be a little bit surprised that I don't think many people will see coming. So first of all, in the background of this slide is an electromagnetic wave. So what does that mean? It means that you have an electric field and a magnetic field perpendicular to one another, propagating in a direction with a velocity v that is perpendicular to both the electric and magnetic fields. So keep that in mind, as it's important and explains really what electromagnetic radiation is. So first of all, what is spectroscopy? Spectroscopy is the study of the interaction between electromagnetic radiation and the matter that it encounters. So one of the biggest or most prominent effects, maybe if you've taken a physics course, you've heard of it, is the photoelectric effect. It was sort of an experimental outcome of inducing electromagnetic radiation on the surface of a material and observing electrons being ejected and not understanding why or how. Albert Einstein, in one of his papers in 1905, collectively known as the Annus Mirabilis, the miracle year, sort of came up with a model for why this happens, suggesting that the energy levels of electrons are quantized or discrete. One of the applications of the photoelectric effect is in spectroscopy. One example is XPS, X-ray photoelectron spectroscopy, where same deal, radiation is shown through a monochromator to create, light or radiation of a single frequency, and the photoelectron is emitted. Okay, so let's go over, an overview of some different spectroscopic tools before we get into FTIR. So maybe if you've taken an organic chemistry course, you've heard of some of these: NMR, IR, UV-Vis, that sort of thing. One of the biggest differences is the wavelengths that they scan, but also qualitatively, what are they perturbating? What are they sort of manipulating? With NMR, for example, it's the magnetic field. But with IR, you're really reading into functional groups and their dipole moments. So it's very interesting. And I suggest that you read up on these other methods, but today we're going to be talking about IR. So maybe the figure on the left looks familiar, and maybe the figure on the right looks familiar. Most people are familiar with the figure on the right, at least in everyday practice. That's the caffeine molecule. And now that I'm looking at it, it actually looks like a purine, one of the bases of DNA, the genetic code. But anyway, if you look at the figure on the left, each of those peaks actually represents a functional group, and a functional group is a structure, made up of atoms that has predictable reactivity and usually some importance. Usually they're used and manipulated by chemists or biology or nature, for that matter. And IR can help sort of point those out. So, for example, these peaks around 3000 here typically correspond to the methyl groups, the CH3. It'll refer to the CH stretch, or 1660 refers to the ketones, these C double bond O's with R groups attached to them. And so you can already see how, the alkene is somewhere in there. I forget where alkene comes out at. But you can already see where this is extremely useful for chemists, biologists, all scientists that deal with molecules, basically, as you can get a qualitative characterization. So briefly, let's talk about how it works. You shine infrared, electromagnetic radiation. You excite these molecules and you see at what wavelengths do they absorb. These wavelengths or frequencies are unique to the functional group, and they only measure the functional groups that are antisymmetric, meaning that they have a dipole moment. So you can see here that there's no net direction, so to speak, these hydrogens are heading in. With a symmetric stretch, they're kind of canceling each other out. If you imagine them in a linear fashion, as vectors going in opposite directions, sort of canceling each other out. This is really interesting. I don't think they usually teach you this when you learn about IR, but you might have heard of the principal quantum number in general chemistry, which describes the energy level of an electron. And that's shown by these thick black lines here. But what you might not have heard about is these in-between energy levels. So these are still discrete, it doesn't go against anything Einstein proposed in the explanation of the photoelectric effect, but they exist in between, and they exist, there's many of them, and they exist with very little spacing between one another. These are rotational and vibrational energy levels. So a molecule rotates and it actually also vibrates. Actually, even at absolute zero, that's one of the myths that a molecule has no energy at absolute zero. That's wrong. And so, anyway, so that's actually explains why you have these peaks, but that you have this continuous curve, because that continuous curve is describing all the little energy levels that the electron could have been promoted to and absorb frequencies at, light energy at. And these peaks sort of represent, I guess, like dips, like, highly probable areas, usually at the principal quantum number in terms of energy level, where this light can be absorbed, as the light is usually at a discrete frequency. Anyway, so let's get into this paper here. I thought it was interesting to do something like this for the educational segment, where they actually used FTIR along with machine learning to help diagnose people with ovarian cancer sooner. So one of the big problems is that testing for ovarian cancer, one of the ways they test for it is for serum levels of a protein known as CA125. But this protein is, this exists in, benign ovarian pathologies or other, pathologies like endometriosis. And so it becomes hard to differentiate between ovarian cancer and something else. And so, the scientists in this paper tried to solve that problem, or at least head in the right direction. And it's very interesting. At the top right here is IR spectra, and they show, the different frequencies of different functional groups of lipids, proteins, those sorts of things that are, what would you say, characterizing of somebody that could have ovarian cancer. That's from the data that they collected in the study, and they had very good success. Serum blood samples had the highest sensitivity and specificity and accuracy compared to urine and other blood fluid levels. And other bodily fluids. So you had sensitivity at 76%, specificity at 98%, meaning that if you get it, if it says you got cancer, or it doesn't, the odds of it being wrong or a false positive, false negative, type 1, type 2 sort of error are very low. And the accuracy is 94%, which is great. And so, they used machine learning algorithms. This is sort of a plot that visually describes how they did this. With a 95% confidence interval, they were able to actually, even with healthy patients, they were able to see who's had past chemo treatment and who hasn't, which is fascinating, I thought. There's so much that you can get from data. So how is this useful? Well, obviously in diagnosing patients, scientists might find it useful because, well, if it's in the urine, if it's in the blood, these compounds that they're picking up are probably unique metabolites of cancer-causing or just cancer-involved pathways within the body, whether it be signaling pathways, metabolic pathways, who knows? I certainly don't. But maybe scientists can use this to sort of backtrack and figure out, how does this cancer work? And then therefore, sometime down the road, how do you treat it? But for now, it's amazing that they can sort of catch it earlier. Anyway, if you want any further reading, there's the paper. It's very dense, but I highly recommend it, and there's a lot you can get out of it. Here's some more on IR if you're interested. There's a lot of physics behind it, so if you're more in the physical sciences wave, I would definitely recommend giving it a read. I love Chem Libre. I think I've probably shouted them out in another episode, but whatever. Anyway, guys, I hope you had fun. I hope you learned something. Just remember that you've got potential.